started at 6 p.m. yesterday. And right now we're doing a quantitative transect. That is a sort of a straight line with using a specified parameters. Oh, look, there's a dead heterocone, which is a sponge. That one that looks SID-y, looks like a big SID. And we're off California here, off in the area off Morro Bay, in an area called Santa Lucia Bank. And we're doing a transect so we can compare different areas. Trying to sample with photographs in the same way in different areas so we can do some counting of the biota in the region and compare it to other transects where the photographs were collected in the same way.
Roger.
So Nav, will you tell us when we've hit our 200 meter mark or our time limit?
Yeah, so we're at 50 meters. We're almost at our time limit, so we're just trying to get that last 50 meters, get the Niskin and pop out.
Awesome, thank you.
So we're running out of time on this dive a bit, so we're trying to finish this transect, get the full 200 meters that we like to run as a standardized transect. Then we'll take a water sample for assessing the DNA in the water.
And we will.
Meredith Everett, who's one. Go ahead.
Sorry, yeah, we will end up pretty much on that ridge, on that local max, so we will have explored that portion.
Great. We're trying to get up to the top of the ridge so we have a really good idea of what this whole area. Oh, look, there's a crinoid walking away, I think. Is that it?
No, he's swimming.
Looks like there's going to be a pretty good ridge rise coming up.
The crinoids look like they're attached to the rocks, all those feather duster looking things, but they can take off and swim around and find a new place. We just saw one of them doing that.
All right, so this is 200 meters if you want to.
Roger that.
I'll stop the show.
And that's the end of the ridge. Jumping off into oblivion.
So shall we take a Niskin here?
Sure thing. Any depth you want to do this one, Kim?
I think we just want to have a sample in this area, not in the water column.
Roger.
Because this is for eDNA.
Roger that. And yeah, the ship moves over with.
Yeah, I can back us up if you need to.
No, it's fine. It's all good, Ben. No worries. Turn on the porch light. Mary, what's the Niskin we were looking for?
Niskin 3.
Niskin 3, roger. And what's going to be our recovery heading?
I'll check in, but I think it's going to be about 290 like we're at.
Okay. Nice. Up.
So for those of you watching at home,
And then, Kim, you
you
want to
see
take one of the plates from the front too and drop it? Let's do just one plate. So maybe the one on the right there.
And you guys just took an Niskin, right? Or two?
Yeah, we just took the Niskin.
Okay, so that's...
Niskin number 3.
so the arm there just pulled that blue loop and triggered the closing of a bottle to take a water sample. And now, in order to help make the ascent easier of the ROV, looks like they're gonna just cut that bit of rope around a weight, I think on the front of the vehicle. You can see the manipulator there.
Data, just to check that Niskin was 18, sample 18.
Yes, sample 18.
Thank you.
Did we want to take any Niskins in the water column?
Yeah, we can. Oh, sorry, Ben, that was to Mary. What were you saying?
Yeah, they're going to keep it at 290.
290, roger. And which way is the swell coming from?
Well, I know our wind is 322, and we're our current's 266, so I think it's probably 322.
That clears out the workspace. Yeah. All right. So I'm going to string us out now, and then be all good. So Argus will be at 290.
No problem. Thanks. He
What was
needs
that?
a little time to get the swell. He says he'll get back to us in 20.
Okay, roger that. Well, we can plan for 290, and then
Yeah, I think that's going to be it.
Nice. Nice.
Ah, I didn't realize you didn't cut it off. You just lifted it off.
I think that'll help.
I'm going to switch us to USBL.
Sorry?
I'm going to switch you to USBL.
Sure.
So just trying to get this slurper tucked back into its home. Just finishing up this dive on Santa Lucia Bank.
Want me to start coming up? Yeah, maybe try rotating a little bit and see if you can finesse it in there. Sometimes I have to like rotate it into the quiver.
So for the Niskins, can we do 700 meters, 200 meters, and 50 meters?
Sure, in depth.
Yes.
Great.
Thank you.
No, no worries.
What is the Nautilus plan after this dive?
Science, are you still there?
Still here, yeah. Just curious what's next for you guys after this dive and sample processing.
Right. As best I know, we are diving again at 4 o'clock this afternoon. I believe that's still the plan.
Okay, Lisa's saying you're heading back to Davidson Seamount.
That's correct, yes.
So there's always cool stuff to see there.
Yes, lots of octopus.
Always a fan favorite.
Erica, can you hear me? I'm having a little headphone problem right now.
I've got you.
Okay, thanks.
So we are finished our dive on Santa Lucia Bank and have begun our ascent back to the ship. We'll be taking a few more samples on the way up and seeing what we see as we move through the water column.
And did I hear we're diving again at 4 in the afternoon today?
It sounds like that is the plan. They're going to transit back up to Davidson and do one more dive up there now that the weather has chilled out a little bit.
I should look at the updated watch list and see if you and I are on at 4 or not.
That schedule is blank. I'm guessing it's going to be the Monterey folks.
Yeah, I think it is. Did you look already?
Yes. Yeah, Lisa's confirming it's the Monterey crew, and we will be in the science chat.
Okay. Sounds good. Not as much plankton in the water column as we see sometimes. But we might still be a little too deep. I'd like to see another one of those blood belly ctenophores.
But it does look like we'll be back on Santa Lucia starting Sunday afternoon for another dive to in this proposed new sanctuary.
Great. So that's you and I back on the 4 to 8 again, correct?
Yes.
Well, that should be fun. We'll see another spot on Santa Lucia. We're at about 960 meters now, ascending at the end of the dive. We just saw a little red mice that go by. A lot of things in the deep sea are red because red light doesn't penetrate in this deep in the water column, so it makes them sort of invisible. So our next dive in this area will be on Sunday. I think starting in the afternoon. We're going to Davidson Seamount now after we finish up this dive, get the ROV all on deck. We'll go to Davidson Seamount and then we'll come back to this area on Sunday and dive a area we're calling the escarpment. I've got everything all straight in my head. Is that what you think, Erica?
That's what it appears like on the schedule, yes.
Sorry, I've got so many windows open on so many computers, I can't quite get to the watch list right now. For those of you at home, we divide up the roles. We call them watches. And everybody does their part in 4-hour shifts. And some of us are talking to you live, and some of the scientists are in a chat room helping us conduct these dives and giving us more information about what we're seeing and also what we need to do and collect. I'm hearing from Lisa, yes, that our next dive here on Sunday will be very deep, 3,800 meters to about 2,800 meters. Or we might be at a scarp area that's at about 1,000 meters. So join us again at this afternoon where we'll be I think, on Davidson Seamount, and then again on Sunday afternoon, where we'll be at Santa Lucia, back in this region again. And again, for those of you at home, there may be some times during both of those dives where we will be able to take some of your questions live. I think that may be on the schedule, but we will see. It's always fun to hear your live questions. So interesting, and so many interesting questions from the public listening in. Fortunately, we're not taking questions now, but we could be on one or both of those dives for a short period of time.
So I have a question. Do you know if we're sampling through an oxygen minimum zone in this area, or is the interior pretty well oxygenated here?
Well, I'm looking at,
Our
yeah, I'm looking at the data on the sensor right now, and it is not really, I wouldn't call it hypoxic here. It's, I don't think we're going through right here at this point an oxygen minimum,
Yeah,
but it's
might be a
pretty
lower
typical of, yeah, hypoxic means lowish oxygen, but it's very typical off the West Coast
right?
to have a pattern of having an oxygen minimum zone in the water column. The surface water should be really fairly well oxygenated. And then as you go down deeper, you'll hit a zone which is the oxygen minimum zone, and then you'll start having oxygen go up again, sometimes nearer the bottom. You up near the surface, it's well oxygenated usually because of phytoplankton are photosynthesizing where there's light, and they're producing oxygen. What you see is hypoxic areas or lower oxygen further down because some of the plants and animals that are abundant in the surface waters, they begin to decompose deeper, and that takes up oxygen and produces an oxygen minimum.
Yeah, cool. I was noticing there was a lot of plankton all of a sudden. I was wondering if we were at one of those oxyclines, lower one. But whenever we're on the West Coast, I'm always wondering, is this a true OMZ?
Yeah, to tell you the truth, it's hard for me to plot in my head
Yeah. What's a
the
gradient?
oxygen data.
Yeah.
Yeah. So, and I haven't been watching it well enough to see that. I'm trying to see if I can get a better idea. But your plots of, you're plotting, oh, there we go. I'm moving down here. Trying to look at the plot of oxygen saturation here over time.
We're going to want a Niskin in 50 meters. 700, 500, 200.
Does look like the
700.
oxygen is going down as we go up a little bit here.
700, 250.
Okay, roger. I think just knew.
As far as I can tell, the oxygen is continuing to go down here
Yeah.
pretty
Make
steadily.
a bet on where the minimum is.
Yeah.
I'd say 350, 400.
All right, we'll see. I'm tracking it now.
Really going off nothing but a guess.
What's your guess? You said 750?
I'm gonna 350 or 400.
350. Okay,
Maybe a
I'm
little
gonna
shallow.
say 5.
Yeah.
I'm gonna say 500. So we'll see who.
I'm gonna say
Still going down.
499. No.
All right, Mary, we're going to take your 700 meter sample here.
All right, great.
So you see the manipulator, for those of you at home, is going over and it's going to pull one of those colored lanyards and close a water bottle and
And we're
take
going for
a
4?
water sample here.
Yes.
Okay.
I think we're taking a sample at 700 and then 400.
It was 200.
700, 200, and then 50.
700, 200, and then 50.
Nice.
Got it?
That is precisely at 700. That rocks.
And just to confirm, Mary, that was 019.
Yes.
Got it.
Nice, Kim. Sorry? Oh. Yeah, that's all right. Oh. Oh, no worries. Yeah, that's correct. All good. It's our humidity in the bottle now. So nice. Knock on wood.
We're at 600 meters now, and it looks like, to me, like the oxygen is coming up a bit here.
Ah,
Yeah.
so you win. Way closer.
Well, the oxygen data are a little noisy right now, so it's
Yeah.
hard to tell. Looks like they're coming up, though. Looks like it's coming up now. So 600, that's actually a little deeper than I thought it would
Yeah.
be.
And it didn't bottom out to really hypoxic conditions or anoxic.
No,
Severe.
no. Yeah, it's coming back up now. Pretty steadily.
Yeah.
The oxygen data are a little noisy on this scale, so it's kind of a little hard to see what's going on. But it's shooting up pretty fast right now. And what's hypoxic and what isn't?
I think
we know what anoxic is,
yeah.
But hypoxia is in the eye of the beholder.
Yeah, exactly. It's like, what do you care about physiologically or otherwise? Seems very relative to the study. Or
so, yeah, a lot of these organisms are tuned to dealing with lower oxygen conditions. So to them, it's a walk in the park. When you worry is when things are anoxic, when there's no oxygen. And that's what people talk about when there's big die-offs in surface waters, or it's when oxygen conditions are something unusual that animals don't see. So very low oxygen in waters where they're usually very oxygenated, then those animals have a very difficult time.
When there's even concern that in the persistent oxygen minimum zones where you have this depth band where there's low oxygen, you have migrators, some of which come from below that have to migrate through that. And so they only have to deal with it temporarily. But if they're going to like a fixed depth or, you know, something light driven, that if those expand, that depth band expands, they're going to have to endure longer durations in kind of suboptimal oxygen conditions as they migrate. Ooh. some jelly. It's a curious question.
Exactly. So when they, and that's one of the physiological questions is, what techniques do things use to get through the oxygen minimum when they're vertical migrators?
Right.
You know, if it's short, they can, how can I put it, physiologically hold their breath, basically. But, you know, when it expands or is unusual in either in size or, well, spatially, when it's not where it's supposed to be, it kind of knocks the organisms for a loop. Yeah. They're just not set up for it. So those who normally see an oxygen minimum zone, they have ways of dealing with it. But when it's a surprise, even what would be considered not particularly low for animals who are ready for it, it can kill animals that aren't ready for it. Let's see what depth we're at now.
We're about 500.
500 meters, yeah. And the oxygen's really going up fast now. And I would expect to see this go up steadily all the way to the surface. Right. We'll see. So we're at over 1% saturation now, and I would be imagining that we'll see it up at 80% saturation at the surface.
Sometimes you can get over 100% with all the phytoplankton, right? So super saturated.
Yeah, it can. Yeah. You can get oversaturated water at the surface because the phytoplankton is, especially during the daytime, you know, when they're really, when the primary producers, the phytoplankton, are really photosynthesizing, you'll see that.
Yeah, I've seen some neat kind of high resolution sections where you can see that diel pattern in just the surface oxygen, just from that respiration and photosynthesis rhythm. It's interesting to think about.
It's really cool.
Yeah.
Okay, we'll guess the saturation at the surface. I'm going to guess 80.
I'm going to guess, I'm going to guess 95.
Ooh, okay.
So I have no a priori insane chart what the profiles have looked like around here. Like the pyrosomes are definitely not contributing to that.
Are we seeing any pyrosomes here?
Didn't we see them on the way down? Yeah.
Yeah, we
I've
saw them
seen
on the way down.
Yeah, seen them for a lot of dives. Me and Jess were talking about them and how it's interesting that they're, of all the invertebrates, they're the most